Alright, we are picking up with question 7 on the practice biology air test. Looking at question 7, we've got a pedigree. Remember, pedigree is kind of like a family history for a trait. And we're looking at, these are mice, and they have a trait that is sex-linked. Remember, sex-linked means it's on the X chromosome, so that females inherit 2 copies of all those genes. Males only inherit 1 copy, so males and females inherit these things differently. So the question is, this trait, which is a short tail trait, is dominant and it's sex-linked. So anybody who's shaded in here has the trait, and your job here is to find out what, if female number, or organism number 6 here, mouse number 6 is female, what is their genotype? So we need to know what letters they're going to inherit. We could walk all the way through this. I guess we could do that. This daughter got an X from dad and an X from mom. We know dad can only give 1 X. He does not have it. He's got the long tail trait. He's got X with a little b. Mom has X big B, at least one of those, but we know because mom's having a daughter that is long-tailed, she must also have the long-tail trait. Remember, short tail is dominant, so if she was X big B, she would pass on one of those copies, and this would be a female that was short-tailed. So she must be heterozygous. So this female is homozygous for the recessive trait. That's why the recessive trait shows up. The male has the dominant trait. He's got the big B, and he's also got a Y chromosome, so he's only got 1 copy of that big B. So what are we going to pass on here? If we have a male gets an X from mom and a Y from dad. So mom's X's are all recessive, little b. Dad's Y doesn't do anything, so these guys are going to be, all the males are going to be long-tailed. A female, on the other hand, will still get a recessive trait from mom, but dad has this dominant X allele. So since mom and dad both give X's, dad's X is dominant. This female will have a short tail. She's going to have the dominant trait. So what is her genotype? Again, from mom, she's going to get the recessive copy. From dad, the dominant copy. So she's going to be X big B, X little b. That's going to be answer A. Again, she's going to be a female. It says that in the question. So we know C and D aren't right. It was really just between these two. So she's going to get a dominant allele from dad. Letter A wins. Next up, this one is asking what type of transport each of these molecules would need. So the key is the little circle ones. These ones are oxygen. This is glucose. This is sodium. These are ions, so these have a charge. So in the prior video, we discussed this. In the first video, the first section of this test, we talked about charged things don't go through the membrane very well at all. Things that are really small and uncharged go through the membrane really well, and things that are sort of bigger, maybe they have a charge, they have a little tougher time. They might need some help, like with a protein channel. So what we're looking at here is oxygen. We have to pick one of these 3 goes in each spot. So the one that goes through the easiest here is oxygen. And so oxygen is just going to be able to diffuse right through the membrane with no help. The next, let's do this one next. This actually, if you look at the arrow, we're actually going through the membrane the wrong way. There's a lower amount here. There are fewer sodium ions down here, and we're pushing them to the side that's already crowded. If we're going to push against that normal flow, remember the normal flow is from high concentration to low. In order to push against that flow, we need to use something that uses energy. So we're going to use a pump. We would call that a sodium pump. It pushes sodium across the membrane. So that leaves this one, and that makes sense because glucose is a bit bigger than oxygen. It's got some polar regions, so it does not go as nicely through the membrane. So glucose will need a channel, and this is what insulin does. When your blood sugar goes up after a meal, insulin basically activates these channels so that all the sugar that's in your bloodstream can go into your cells so they can use it for energy. Alright, so that's where those should line up. And next question. Next up, we're looking for which prediction is supported by what we see if we looked at gel electrophoreses. Remember, these are where we see the different bands. The different bands represent different sizes of DNA. And so the question is, and for electrophoresis, remember, if they were to match, if the two sets of bands match, that means that the section of DNA we're looking at probably is the same DNA, meaning it's from the same individual. And if it matches mostly, that means they're probably related, like a sibling or a parent. And if they don't seem to match at all, then they're probably unrelated to each other. This is like how they do paternity testing. This is how we test evidence from a crime scene. And so based on that, which of these statements, which of these predictions seem to be true? So letter A, cell 1 from individual A will have an identical banding pattern to cell 1 from individual B. No, typically, no. These are two individuals of the same species. We're assuming they are not twins, so they are different. Those banding patterns should not match, so A doesn't work. B, cell 1 from individual A will have an identical banding pattern compared to cell 2 from individual A. This is the same individual, and guess what? Every cell in your body has the same set of DNA, which means you're probably going to have bands that match. So this one is probably going to be our answer. Let's look at C and D just to make sure. Cell 1 from individual A will have a different banding pattern compared to cell 3 from individual A. 1 and 3, same individual, should have the same banding pattern, so different would be incorrect there. And cell 1 from A will have a different banding pattern than cell 4 from A. Again, same organism. It should be the same type of banding. So, which one did we say was right? That one. Identical banding pattern to cell 2. These guys should match. How do prokaryotic cells differ from eukaryotic cells? Again, the big one. Prokaryotes don't have a nucleus. Their DNA is in the cytoplasm. Eukaryotes, they have a nucleus. That's where their DNA lives. And then the other one for prokaryotes is they don't have membrane-bound organelles. So, unlike eukaryotic cells, prokaryotic cells do not have any chromosomes. No, they definitely have chromosomes. Unlike eukaryotic cells, prokaryotic cells do not have a cell wall. Wrong. Prokaryotes do have a cell wall. Some eukaryotes, like us, animals, don't have a cell wall. So that's actually kind of backwards. Unlike eukaryotic cells, prokaryotic cells do not have any membrane-bound organelles. Now we're getting somewhere. Again, eukaryotic cells do, prokaryotic cells don't. So this one sounds correct. Let's check letter D just to make sure. Unlike eukaryotic cells, some prokaryotic cells move using cilia or flagella. These guys do, but so do these guys sometimes. Some eukaryotic cells also have these things to help them move around. So C is the right answer. By the way, cilia is like when you have a bunch of little hairs that act like little oars, like for like a Viking ship, or a bunch of little hairs swim you along. And a flagellum is a big tail, one big tail. Instead of having a bunch of little hairs, this is a big longer tail that kind of whips around and acts like a boat motor. So letter C is the right answer for that one. A mutation occurs in DNA strand. We see the original strand GGG, GAG, TTA, and we end up with GGA, GAG, TTA. We want to see what the difference will be in the amino acid sequence. So remember, we read 3 letters at a time to decide what amino acid will be used. So reading those letters, this is DNA. We don't read it from DNA. We go from DNA to RNA first. So GGG actually pairs up with CCC. So normally we would expect this protein, this amino acid, to be CCC is proline. Now we're going to have CCU. Remember, we get U when we change over to RNA. CCU. So CCU is still proline. So if we look at this, proline is unchanged. No, that's not it. What did I miss? Alright, folks, I'm back. I'm not sure what's going on there. I thought we were going to focus on just the one place where a mutation was, but it was actually asking about more parts of the chain than I expected it to. So the answer to this should be that all parts are unchanged. The only thing that would have changed, again, GGG, that makes CCC, that's proline. The change was GGA, that makes CCU. CCU is still proline. So that part, proline, is unchanged. And then asparagine is actually the third one here. TTA would make AAU. That's asparagine. And then it's still AAU, so asparagine is also unchanged. So I'm not sure why I included all that. Honestly, it was only that one part. Should have just asked about it. But anyway, C still works as the right answer here. C is correct. Next up, we're drawing 4 cells. Which one's a prokaryote? You guys might notice a theme. We see a lot of prokaryotic, eukaryotic questions here. Looking at these cells, this one's probably a plant cell. We see a bunch of organelles inside, so it's a eukaryote. There's a nucleus, it's a eukaryote. Here's a nucleus, that's a eukaryote. This one lacks a nucleus, that's a prokaryote. Alright, and that is, we'll stop there for this video.